actually. This is a slide, very nice slides from Bin Yang. It's one of our partners in the project, in the work. Just represent what is happening with tal effector. So tal-C, for instance, this effector that is in African populations, recognize just 20 amino acid, 20 nucleotide in the DNA of sweet 14. Have a specific target sequence, and we design guide RNAs in the construct that target this region. These guide RNAs, of course, was recognized by the Cas9. The Cas9 is a nuclease that cut the sequence. This sequence is repaired by a system that is not specific in the plant, at least in higher plants. And this non-specific reparation will introduce a set of mistakes. And this is the base of our strategy, that the fact that by chance there are more mistakes than correct reparation give us a lot of room to play with this tool. Of course, one beauty of this approach is that the tDNA insertion happen in one of the chromosomes, and it's highly likely that the target region is in a different chromosome. So you can easily segregate these two plants and continue the T1 with a tDNA-free plant. In here, what you observe here is some of the results for a couple of the regions. What we did is actually target 5 different regions at the same time. I'm just showing 2. In yellow, you see the wild type sequence, the original sequence, and in red, if you can see the red here, you have several results or sequence that we obtained by CRISPR in that region. So for instance, 1A in introduction, couple of T's, 1T. Here in the different sequence, we have 2 A's insertion. We have a deletion of 7 base pairs in here, and so on. So we hope that accumulate the most number or a good number of mutations and decide what mutation can give us what we need. And what we did is that enough difference in the sequence to block the binding, but what we don't want is big deletions that may affect the promoter or may affect some regulatory element that is close by. So we need to be very careful about this. And this is what we did. This is a summary here of the 3 genes and the 5 locations using different regions. And we select these plants, and T2 or T3, we inoculate with different strains of XOO. These strains are carrying one single TAL effector for each suite. So what happened here is that we clip inoculate, so inoculate in the top here, and the bacteria move down. And as you see, the bacteria is taking over the tissue. However, in the plant that has been edited, the bacteria is inoculated in the top, start to the infection, probably is very good at the beginning, but soon after the sugar run out and the bacteria die or starve to death, basically, and the infection stops. So you can see the same here in the greenhouse experiment this time, where the infection is almost none in the genome edited lines compared to the wild type. Here are the plants in the greenhouse. You can see they are very nice and happy plants. However, not everything is good. As you know, we are using genome editing. We are using transgenesis. So some plants we expect that there are a trade-off, different targets or targets that you are not expecting. And also the transformation itself can introduce some mutations or some changes in the structure of the genome that can change a little bit the plant. And we detect that when we did the agronomic trait of these plants, what we measure is different agronomic characteristic of the set of plants to see if they have a trade-off effect with the wild type. Here's the wild type, and in most of the cases, we have similar measurements for yield, fertility, plant height, and so on. We only find one plant out of 20 that shows a little bit less fertility than the rest. And this plant in general is a little bit different. You can see here the panicle is a little bit different, smaller. And this plant, sorry, is this one actually. And you can see that immediately kind of glow as a different plant. At the beginning, we thought that was a contamination, a different plant that we planted there, but no, after the analysis, we realized that this same plant, good mutations because it's resistant, but somehow we hit some part of the process and some clonal variation during the transformation changed the plant a little bit. But yeah, if you want to develop products, you need to go to multi-location trials to understand what is the effect of the field, what is the effect of the environment in the plant, because you don't know what happened when the plant gets stressed by drought, by salinity, or other stress agents, how that plant will react. So what we are doing at the moment, we're working with our colleagues in Colombia, in Burkina Faso, or in the Philippines. Also, we start working in Uganda and India to try to test those plants in a multi-location trial to assess exactly that. What is the effect of the mutations in the real life situation? So far, so good. The results are good. Everything is ongoing. This is our first experiment. Few photos of our first experiment in the Philippines. The plants look good. Look here, look healthy. The feeling looks okay. Here is Paolo. He's the lead of this project, or this part of the project. He also is good photographer, so we are making a record of that. Also, Jenny Lu have been instrumental in this this part, also doing the transformation and so on. I will change a little bit topic, not topic, but disease. And I have one slide. This is blast. This is a different pathogen. It's called Magnaporte oryzae. It's a fungal pathogen from leaves and panicles, and have a similar interaction that XOO, but is also a very big problem. What I want to say here is that there's many genes, resistant genes, that are used to control these fungus, and most of the genes are NBSLRR, basically cytoplasmic receptors. And what I want to say actually is that we are also doing some research on resistance based on starvation mechanism. We are targeting some of these sugar transporters, not sweet as the XO, but other type of sugar transporter that allow to have this, to block this process of importation of glucose and fructose, to block the access of the Haustoria to the nutrient. So I won't talk about this, but probably in the future, I will be happy to discuss with you guys about this. So this is my final slide for this part, and I just want to touch briefly on the actual landscape of genome editing around the world. This is a nice review from Sarah Smith in 2020. I fully recommend you to go through that. It's very nice to read. But this, in summary, is here all the green countries already have or have no regulation for genome edited products as GM, compared to the red countries, which mostly Europe, which have a stable regulation of genome edited product as GMOs. But the most interesting part is all these countries that are in yellow that have now a regulatory process where GMOs are separated from genome edited plants. And this is very good news because these countries are in the front line of research, and probably soon or probably next year there will be some news coming from those areas. So you just keep an eye on that. So to continue with our strategy, the second point that I want to talk with you is about the pathogen itself. Once we have the right gene or the right number of genes that we want, we need to understand the pathogen and try to monitor the pathogen as much as possible in the regions that we work. So we are lucky because for XOO, we have a lot of information. As you remember, this calls bacterial leaf blight. And these are the locations of the genomes that we have been sequencing as a community. There's around 430 genomes across all Asia and across the last 40 years of agriculture in Asia. So we have been capturing this diversity as much as possible. And what we know so far is there are 3 big groups, and there is 15 populations. And 15 populations, each population have its own evolutionary trajectory, its own history, right? When I refer to history, that's the recombination process, the selection process, the way they accumulate mutations, and the way they interact with the host. So these 15 populations are a key number for us to try to monitor and to understand what is happening with each of these populations. As I mentioned, for these 15 populations that are represented here, we know also the phenotypes, and we know, or we try to understand what are the TAL effector repertoire. So this is telling a lot of how the populations evolve, but how also they have prepared for interacting with rice. And we know, as I mentioned, a lot of these populations. I won't go into each one by one, but each population have its own kingdom and its own way to be controlled, right? For instance, here in South Asia, we have population 4 to 6, and is easily be controlled or can be controlled by one resistant gene that we know. But other populations at 7, 8 probably need 2 genes to be controlled, and so on. So we have this information, and we can modulate the breeding program, and we can modulate the response to that pathogen based on this. We also know that all these populations together globally can be controlled, or can be controlled by a combination of sweet genes. Sweet genes, basically the products that we developed in the previous part, either by sweet 11 or sweet 13, 14 together, or the 3 of us together. And why I talk about population? Because remember, I described how we need to understand the genomic context and the way the populations evolve and the way they carry the genes to develop very simple markers, very simple tools that allow us to fast track this pathogen. So we develop SNP markers that are stable and can be detected high throughput in the leaf. So you just need a piece of leaf in the field, put in a Epender tube, send to the genotyping service, and that's it. So you can have all this information in a couple of days, right? If you remember one of my first slides, to do this in a traditional way take years to try to isolate, understand, sequence, or do many tests. Now we can reduce all this time into a very small fragment of time that allow us to respond faster. If these go to the right person, the right breeder, or the extension service, we can contain the disease in time. And as you see here, we were able to, in 2019, we were able to genotype or profile 1,000 outbreaks across Asia, from Pakistan to Indonesia and in between. So in a couple of weeks, we have a good sense of what is happening in Asia. And definitely we can respond. So what we want is to articulate this and make this a process for each season. So each season we can monitor what is happening with the population in each of these countries. Right now, I will jump to BLAST. Remember, BLAST is a fungus. The genome is way bigger, 10 times bigger. Is similar to a rice genome, actually. And to summarize again, the interaction is very clear. The fungus interact with the plants using this Haustorial that will secrete effector genes, as I mentioned before, and the plant have the resistant protein that detect these genes to trigger a defense response. In the breeding program, we have already several genes we call PI genes. These resistant genes are already in the breeding program. So if we know that these resistant genes detect effector genes, we want to do the same process that we did with XOO, but to BLAST. This time, we want to target effector proteins using our markers. And why? Because knowing the frequency of effectors help us to understand how effective is the resistant gene. So, for instance, if a effector is 100% present in the population, like here, that means that if I deploy the resistant gene that interact with this effector, it's highly likely that this resistant gene will be effective, right? However, if the population delete the effector or have almost low frequency, that means that your resistant gene will be not so effective or not effective all the time. And we know that this frequency change with time. This is an example of the Philippines. In the old collection, we detect this particular effector ABRPI-ZT at half of the frequency. However, by the new collection several years later, this effector almost disappeared for the same region in the Philippines. So we know that there's a dynamic. So we need to make sure that we monitor that dynamic in time, and of course, more effectively. But of course, Philippines is one part of the puzzle, and what we are looking is the big picture. So to mimic what we did with Santomonas XOO, we went to a similar strategy in which we acquire genomes from all over the world. Our collection from Africa was represented here. We did collections in 13 different countries in Africa, and also in Asia. But of course, we also use genomes from other publications. And just to give you a summary of the situation, BLAST originated in Southeast Asia. And this was several hundred of thousands years back. And just recently, the last 100 years, probably colonized Africa, probably in the seed system. However, what we find in Africa is a very diverse population. Basically, all the groups that are present in Asia are now in Africa. And these two populations are evolving at different rates and different times. And I will show it. This population was out yesterday, actually. And you can check that here. What I meant here is that when Africa and Asia are having different rates, we measure an index that is called Tajima. And Tajima basically measures the selection process, or the selection process that those populations are being through. And in this case, we know that Africa and Asia have different Tajima values, meaning that they have different selection processes going on. We can observe these values here in a very small fragment of the genome, chromosome 3, I think, of BLAST, in which one of the populations in Africa have different Tajima values to the populations in Asia, and this can be represented or can be represented at gene level. So the gene diversity of these two continents in the same population with the same background pool is different. So that means that something is going on in Africa compared to Asia. This is true for the whole genome, but also true for the effector genes that are our target of interest. And one of the key measurements is the number of effectors. How many effectors that the population have. And when we measure the number of effectors between Asia and Africa, we recognize that there is different number of effectors. And that means that the gain and loss of effectors are also changing and are subject of the interaction. So obviously the hosts in Africa have something to do with this interaction, and the populations are reacting. What we observe here is basically the present absence polymorphisms of all the 178 effectors that we found in the genome. And we need this information. Why? Because, as I mentioned, we need to develop tools or markers that allow us to track some of these effectors. So we develop these markers. At the moment, we are in that process, and we are targeting all the effectors that are being recognized by the genes in the breeding program. So the genes in the breeding program, the resistant genes in the breeding program, have a representation in this set of markers. My point is, whatever we are doing in breeding, we are monitoring in the ground using these markers. So we can advise breeders what to do. If the effector is becoming low frequency, it's better not to use that resistant gene, or it's better to use the resistant gene, and so on. So we are in this process at the moment. This is the final part of the presentation, and I want to talk a little bit on the decision making. So, as I mentioned before, we need a platform to integrate whatever happened with the host on the resistant part, on the breeding part, and whatever happened in the pathology part and the environmental part. So we need a decision platform that allows us to give advice on what resistant gene to use or what variety to deploy the next season. So we create this tool that is called Pathotracer, and is a very simple and very friendly platform that allows decision making on diseases. Again, for a breeder perspective, breeders need to know what resistant gene they need to advance, right? Imagine that a bad decision costs money a lot. So if you advance the wrong resistant gene, then there's a lot of money and resources spent that. And then they need to know this on time. And the second part is, for extension service, you need to know what variety will be resistant, or existing variety will be resistant in that location. So we pretend to develop this tool to, again, democratize this information and try to everybody see the same, and so everybody can have similar decisions. And as a visualization tool, it's very simple. You just at least this is bacterial blight. It's named for XO populations. And we try to represent the dominant population in each region. So if you go here, if you click in one of these or provinces or regions, you will have information like the dominant population, what is the collector, what is the resistant gene that is needed, or what is the variety that is out there that can stop this pathogen, or have the resistant gene profile that stop this pathogen. You can see here, for instance, in the Philippines, if we click in one of the provinces, we have the profile of this province, but also we have the rice accession that can be used in the breeding process or directly in the field to be planted in the regions with other characteristics as well. So basically, this tool is a platform that integrates resistant and pathogen and environment in one single platform that is simple enough that everybody can have an idea what is happening. And of course, there's a lot to do still, but we are working on that to integrate more diseases, not only bacterial blight and blast, but we expect to have virus diseases and other important diseases in other regions. This is the same platform, and this is a visualization from blast. Of course, each pathogen will have its own particular set of of visualization. In here are, remember, the effectors. So if we click in this location in the Philippines where IRRI is located, we know that the frequency of these effector called ABRPIK and ABRPI9 are almost 100%. That means that if you use any of the genes, the resistant genes that recognize that effector, you are safe deploying that variety.